Hello, I am Dr.Ashok Kumar, Department of Zoology, Ganga Singh College, Chapra, running under JP University, Chapra, Saran, Bihar. Today I am going to switch on the topic diseases in fish caused by protozoan parasites. The protozoans are one of the important group of single-celled animal parasites which affect fish. Most of the organ systems of the fish are infected by these parasites, and the majority of the groups of protozoan parasites, they brings death among fishes, so it causes mortality. The important diseases in fish caused by protozoans are as follows. There are mainly two types, but on the basis of the spots on different organs, suppose on gill, white spots which appear on gill, then it will be called gill spot disease. When white spot appears on the scale, then it will be called white scale spot disease. So the following diseases. First is ichthyopthriasis. Ichthyopthriasis means the causative agent is a protozoan, that is Ichthyopthrius multifilus. Ichthyopthrius multifilus. Second is trichotillomaniasis. It is caused by Trichodina and Tripartiella genera. So, among Trichodina reticulata, Trichodina nigra, and the genus Trichodina reticulata, Trichodina nigra. These are the two different species of Trichodina protozoan. And the second is Tripartiella. So three types: Tripartiella copiosa, Tripartiella bulbosa, Tripartiella obtusa. These are the three types of trichotillomaniasis. White gill species, that is caused by Thellohanellus cattleyi. Thellohanellus cattleyi, Myxobolus bengalensis. Thellohanellus cattleyi, Myxobolus bengalensis, Myxobolus cattleyi, and Myxobolus hosadurgensis. These are caused by Myxobolus and Thellohanellus. So, Thellohanellus cattleyi in Katla, Myxobolus bengalensis, Myxobolus cattleyi, and Myxobolus hosadurgensis. So, these are the three varieties of Myxobolus and Thellohanellus cattleyi, that is caused by Thellohanellus genus. And white scale spot disease, they cause white gill spot disease. Thellohanellus and Myxobolus, they cause white spots on the gills, while white spot on the scales, that is caused by Myxobolus mrigeli, Myxobolus sphaericum and Myxobolus rohiti. So Myxobolus mirieli, Myxobolus sphaericum and Myxobolus rohiti. These are the Ichthyopthriasis, trichotillomaniasis, and white gill spot disease and white scale spot disease. These are the protozoans disease. These are the occurring in fishes, mainly carps. Now ichthyopthriasis. It is fly. It causes especially affected by are in the case of ichthyopthriasis disease. It is also called itch disease, khujli hona, itch disease, ICH. So fly and fingerlings of katla, the species affected by Ichthyopthriasis multifilus. Ichthyopthriasis multifilus, it brings itch disease, and here the causative agent, the species affected are the fly and fingerlings of katla, Labiorohita, Sirhinus myrgella in nursery and rearing ponds. So fly and fingerlings become infective. External symptoms of ichthyopthriasis. These are the minute white spots. It causes minute white spots on the skin. These are on the nodular form, white spots in node gant in nodular form. Minute white spots in nodular form inside are visible on the skin, fins, and gills. So these are in nodular forms. They are white spots, diseases which are found on skin, fins, as well as gills. And affected fishes show irritation. Affected fishes show irritation and erratic movement, such as itching. हम ही लोग को itching जब होता है तो हम ही लोग बेचैनी महसूस करते हैं, और such type of itching is also there, so it becomes restless and erratic movement takes place. So there is a tendency of the fish to rub on the sides. यह जाता है, it goes there and it rubs on the sides. So for minimizing, so restlessness and erratic movement and a tendency to rub on the sides of the pond. So on the bank of the pond, and it goes and rubs itself, its body there. It is this has been reported by Gopal Krishnan in the year 1963 and 64. Now effect on host. After effect on host, epidermal tissue changes occur in the area where the parasite is lodged. It has been explained that it remains in nodular form. So on the host, epidermal tissue changes occur in the area where the parasite is lodged. Epidermal tissue changes. The dislodging of the parasite causes formation of epithelial ulcers. When we dislodge, it means when we bring out with any sharp forceps, the nodules, that becomes, we see when the nodules comes out, we see ulceration. Ulceration means ghav. Ulceration means that is blood is coming out. So ulceration, epithelial ulcers. Causative agent. Now the causative agent is Ichthyopteris multifilus. It is this disease, ichthyopteris is caused by Ichthyopteris multifilus, and it has been Pocket in 1876. He reported it, and not only fingerlings of Labiobata, Sirhina, Mrigala have been experimentally infected with this parasite by Tripathi in the year 1955. This parasite was infected, and it was found that after infection it goes and makes nodule-like structure in the skin, in the epithelium or skin, and after removing, we see there is ulceration inside the dermis or skin, inside the skin. Fingerlings. Now, morphology. The mature here, morphology means shape and size. So, epithelial effect on host causative agent is Ichthyopteris multifilus, Pocket 1876. Fingerlings of it is found in fingerlings of Labiobata, Sirinus mrigla, and morphology. The mature tophone's body is circular to ovoid, golakar to ovoid, andakar. So the mature tophone's body is circular to ovoid, measuring 600 to 800 mm in size. It is covered with, so body is circular and circular to oval. The mature tophones of Ichthyopteris multifilus, it is circular to oval, measuring 60 to 80 mm in size, 6 to 8 mm in size. It is covered with 36 meridional rows of cilia. It is covered with 6 meridional rows of cilia. The mouth or cystosoma is present anteriorly at the bottom of the vestibular depression. Mouth is present at the bottom, anteriorly at the bottom of the vestibular depression. There is a depression that is called vestibule, and mouth remains there. Now the depression, macronucleus present in large and horseshoe shaped. Its nucleus, macronucleus and micronucleus. There is the macronucleus. It has the macronucleus present is, macro means large. It is large and horseshoe shaped. Its shape is horseshoe like. So horseshoe shaped nucleus. Life cycle. The fish, the infective stage of host is therion. That is, these therions are migratory. So infective stage of the host is the migratory therions. which infest fish skin or gills, which fish skin or gills. It attaches with the skin or is attached with the gills. Now, once inside, when one therion goes inside the body, inside the skin, what happens? So once inside, it starts feeding and growing at this stage. So now once the therion goes inside the body, in the skin or in the on the gills, on the gills, it goes inside and starts feeding and growing. So after feeding from the fish, it converts, it becomes large, and it converts into this stage called trophons. The trophons, they are large. Therion infects and trophons are large. Now, after taking food from the tissue, its body grow, and as a result, they are called trophons. Now, the trophons, on reaching the size of 1 millimeter, the trophons on reaching the size of 1 mm escapes from the host. Trophons, therions goes inside. Then it takes meal from the cytoplasm of the epidermal or epithelium cells, and it grows. These trophons that become 1 mm in the size, and after attaining this size, it escapes from the host. It comes out from the skin as well as gills, and insists on convenient substrate outside as tomones. Now the trophons comes and it searches a substrate like rocks, like hard substratum, like wood, like rocks. So it attaches itself, and now in the favorable environment, this convenient substrate outside, and it encysts itself within the cyst. Now it is called tomont. So first was the infective stage, that was therion. Now after taking feeding, it comes on, after feeding it comes on, therion becomes large size, 1 mm size, and the therions become large size, 1 mm size, and this therion then comes out from the body of the fish, and it attaches itself with the hard substratum, like stone, like brick pieces, or wood, any hard substratum. Then it becomes, now it attaches there, and now forms from forms an envelope around its body, and this body, it is called tomonts. Now the tomonts, these are now therion, and now it is therions, elongated structure, and therion becomes trophons. Then trophons comes out and it attaches with the substratum. Now it is called tomonts, and these tomonts remains inside an envelope or cell, and they now they divide inside the tomont. It divides into and produces a small tomites. Now it divides 10 to 11 times, 10 to 11 divisions to produce a small tomites. Now the tomites are inside these tomites, small tomites here, 1, 2, 3. After 11 division, so many tomites are inside, and now it breaks the envelope. Now it breaks the envelope and comes outside, and tomites becomes and these tomites now the cyst wall breaks and tomites becomes theriont again. So theriont, first stage, theriont becomes trophons. Trophons comes out and attaches with the substratum and an envelope. It secretes an envelope around itself. Now they are globular rounded structure. These rounded structure, tomons, the cytoplasmic nucleus, they divide 10 to 11 divisions, and then these tomons are called tomites, and these tomites now breaks the envelope and comes out as theriont. Theriont is an elongated structure. This theriont, now the theriont is an elongated structure, and its size is 0.3 to 0.4 mm. So mm in size, covered by 36 to 48 meridional cilia. So these therionts swim freely in these therionts now freely it wanders, swims in the water of the pond, and these are the infective stage, and their therionts remain infective up to 4 days. If they do not find a suitable host, they die after 4 days. But till 4 days it remains infective, and in the meantime it enters a new fish or a new fish or the same fish. On the skin it attaches and goes inside, and after taking feed from the cytoplasmic, includes the cytoplasm, it is lodged inside the skin, and the life cycle is completed. That is all about the life cycle of the Ecthyopteris multifilus. So